cancer has become a leading cause of morbidity and mortality worldwide. In addition to the tremendous suffering it inflicts, cancer is a reason for significant economic burden both to the sufferer and to the nation. 1. Recently, a high-impact report suggested that cancer is primarily stochastic or bad luck because of the accumulation of spontaneous mutations during DNA replication in tissues where stem cells undergo a relatively large number of cell divisions. 2. However, it is widely believed that the environment too significantly influences the risk of cancer. 3. 4. Numerous epidemiologic and occupational health studies support the importance of lifestyle factors and exposure to known or suspected carcinogens in the development of cancer. In fact, it is estimated that 15% to 20% of cancers are driven by infectious agents, 20% to 30% are largely caused by tobacco use, and 30% to 35% are associated with diet, physical activity, and/or energy balance. For example, obesity, ultraviolet radiation from sunlight, alcohol, and many other substances, for example, asbestos, benzene, and radon, also play a role, both alone and in combination, that is, mixed exposures, although relative risk depends on the dose and duration of each exposure and the genetic background of each individual. 5. 6. The microbiota that inhabits our gastrointestinal tract and other anatomic sites can be considered environmental factors to which we are continuously exposed at high doses throughout life. Most of these microbes are commensal bacteria and until recently have been difficult to culture, which has limited our understanding. However, during the past decade, the advent of metagenomic sequencing approaches that combine next-generation DNA sequencing technologies with the computational analysis of targeted, 16S ribosome RNA hypervariable regions, or whole genome shotgun sequence reads has documented the diversity and abundance of microbes at different body sites in a culture-independent manner. 7. 8. It is well known that prostatitis and prostatitis symptoms are associated with an increased risk of prostate cancer. 9. 10. It is probably that bacteria induce a chronic inflammatory state in the prostate that could result in enhanced production of pro-inflammatory cytokines. Both neutrophils and macrophages release pro-inflammatory molecules such as nitric oxide that have the propensity to cause genetic damage which could pave the way for enhanced cell proliferation and cancer. This is particularly interesting in the light of the reports that low-grade inflammation does exist in cancer. U et al. 11. Investigated the type of microbiota in the expressed prostatic secretions, EPSs, of patients with prostate cancer and benign prostatic hyperplasia, BPH, by the polymerase chain reaction denaturing gradient gel electrophoresis method using universal bacterial primers and reported that the prostate cancer group had a significantly increased number of Bacteroidetes bacteria, alpha-proteobacteria, Firmicutes bacteria, Lactospiraceae, Propionicimonas, Sphingomonas, and Ochrobacterium and a decrease in Eubacterium and Effluviococcus compared to the BPH group. Based on these results, they suggested that there were significant changes in the microbial population in EPS, urine, and seminal fluid of individuals with prostate cancer and BPH, indicating a possible role for these bacteria in these two conditions. Cavaretta et al. 12. Recently reported on the microbiome profile of tumor, peritumor, and non-tumor tissues in 16 radical prostatectomy specimens. They observed significant differences in specific microbial populations among tumor, peritumor and non-tumor prostate specimens at certain taxonomic levels. Among genera, Propionibacterium spp. Were the most abundant. Staphylococcus spp. Were more represented in the tumor, peritumor tissues, p0.05. The authors concluded that the prostate contained a plethora of bacteria, which set themselves within the gland with a distribution dependent on the nature of the tissue, thus suggesting a possible pathophysiological correlation between the composition of the local microbial niche and the presence of the tumor itself. Similarly, Shrestha et al. 13. profiled the urinary microbiome in men with positive versus negative biopsies for prostate cancer. They identified a cluster of pro-inflammatory bacteria previously implicated in urogenital infections in a subset of samples. Many species, including known uropathogens, were significantly and differentially abundant among cancer and benign samples, in low versus higher grade cancers and in relation to prostate inflammation type and degree. The authors concluded that there was a prevalence of pro-inflammatory bacteria and uropathogens in the urinary tract of men with prostate cancer.